Hello everyone, welcome to COTSFX. So today in this lecture, we are going to discuss about the HCl formation inside the stomach lumen. So before starting the topic, we should understand few basic terms associated with this diagram, and that is one enzyme which is called carbonic anhydrase. So this carbonic anhydrase basically catalyzing one kind of particular reaction. What type of reaction? We'll understand later on. So it is a carbonic anhydrase. Another term is interstitial fluid. So what is interstitial fluid? It's a fluid present in between two different layers of stomach. So this is one layer and this is our blood capillaries. But these capillaries present in another stomach layer, which is that name of stomach layer is lamina propria. I haven't written that name, but this blood vessel is present in lamina propria. So lamina propria is most vascular layer inside the stomach. So vascular means lots of blood vessels will be there. So this is one particular type of blood vessels. So in between lamina propria layer and our endothelial cells, these are endothelial cells, innermost linings. So in between two layers we have an interstitial fluid. So interstitial fluid is nothing but one kind of extracellular fluid. These are cells, and in the blood capillaries also we have lots of endothelial cells. So this fluid is present outside the cell, so that's why it is interstitial fluid between two layers of stomach muscles. So now next word is apical membrane. So what is apical membrane? Here you see this is our stomach lumen. So stomach lumen means this is the inner linings. This is the inner linings and this is outer linings. This is the outer linings of our lumen. Suppose this is luminal side. So just opposite we have our basolateral membrane. So cell which is directly coming in contact with food particles, that side will be called apical membrane, because chyme is when food particle mix with HCl, then it will be called as chyme. So cell side, our sides of our cell which directly coming in contact with chyme or food particles, that will be called apical membrane, because lumen is present over here. So if it is a stomach lumen, means fluid particles directly come in stomach. So directly come to stomach lumen. So inside the stomach lumen we have suppose one particular cell. So food particle which are coming directly in contact with cell wall or cell membrane. That particular membrane is called apical membrane. So we have two different layers. For example, this is apical side and this is basolateral side. So what is basolateral side? Basolateral side is opposite side of apical membrane. So this is basolateral membrane. So this is stomach lumen. So stomach lumen is inside the stomach, inner linings of stomach. So this is the inner linings. This side is called inner linings. So food particles will directly come and they will make contact with this area, this side of our cell. The opposite of that cell is called towards the interstitial fluid. That side is called basolateral membrane. So these two terms you should understand. Then we have another, these kind of finger-like cells. This finger-like cell is called parietal cell. So what will be happening inside the parietal cell? That is the main point of discussion. So HCl form, the main role will be played by our parietal cell, HCl formation role. So what is the role of parietal cell? Let's get started the topic. So here in case of formation of HCl, first you need to understand that inside our parietal cell we have water molecules. As we know, each and every cell of our body contains water. So this is water molecule, and parietal cell, it's our one kind of stomach luminal cell. So inside that cell metabolic process will be there. So during that metabolic reaction lots of carbon dioxide will be generated, and cellular respiration also this carbon dioxide will be generated. So when H2O means water and carbon dioxide, they make a reaction and they form carbonic acid. So H2CO3 is carbonic acid, and this reaction is catalyzed by carbonic anhydrase. So it's a enzyme. So before initially, as we have told you, that Ca is carbonic anhydrase. So this carbonic anhydrase, it's an enzyme which catalyze these kind of association reactions where water and carbon dioxide make a reaction in between them and form this kind of carbonic acid. So after that, once carbonic acid forms, then it dissociates and form one proton, which is called hydrogen ion, and another anions. So these anion is called bicarbonates. So this is bicarbonate ion and this is hydrogen ion, or one proton. Because our hydrogen plus means electron is not present. As we know, hydrogen contain only one electron. So if we remove the electron from this orbital, then inside only protons will be there. So that's why it is called proton, H plus. Now this is one anion, this is cation. So now let's understand what is the role of this channel. So here you can see that bicarbonates is slowly going towards the interstitial fluid, and once it moves towards the interstitial fluid, from interstitial fluid slowly chlorine molecule is coming towards the parietal cell. So this is finger-like cell. I have already told you this is parietal cell. So everything will be going on inside the parietal cell. So once bicarbonate ion forms, these bicarbonate ions slowly move towards the interstitial fluid, and from interstitial fluid, chlorine ion will slowly come to our parietal cell. Now what will happen after entering of this chlorine ion? These chlorine ion will directly move towards the stomach lumen by using one particular unipotor. This is one unipotor. So what is unipotor, sympotor and antipotor? I'll make another video. But now you can see unipotor is one small channel in which only one directional flow of ions will be possible. Here chlorine will slowly moving towards the stomach lumen. So here chlorine is slowly coming towards the parietal cell from the interstitial side. Now here you can see this chlorine is actually coming from our blood capillaries. So one anion is leaving the blood capillaries, and these bicarbonate ion is slowly moving towards the blood capillaries. So one anion is leaving the blood capillaries, another anion is going to the blood capillaries. So there is a compensation of anions. So when chlorine leaves an ion, to compensate that site, another anion is moving towards the blood capillaries to maintain the pH level. Now here we can see once that chlorine is moving towards the stomach lumen, what is going on with this particular hydrogen ions? Now let's concentrate here. This hydrogen ion is now directly move towards the stomach lumen with the help of particular proton pump. Now this is a positive charge. So when this positive charge is moving towards the stomach lumen, then we need some kind of energies. So these hydrogen ion will be withdrawn inside the stomach lumen with the application of ATP molecules. So here we can write then one particular type of energy we need, that one ATP will be converted to ADP. Then one phosphate molecule will be used to push this hydrogen ion towards the stomach lumen. So when this hydrogen ion is withdrawn inside that stomach lumen, then to compensate that positive charge, our one potassium ion will make entry inside the parietal cell. Now this is a particular kind of proton pump. So the role of proton pump is pumping out one hydrogen ion and pumping in one potassium ion with the application of energy. Here you can see ATP molecule, adenosine triphosphate, and here adenosine diphosphate, means one phosphate is used to push out this particular hydrogen ion out of the cell. So when this hydrogen ion is pushed out, then simultaneously one potassium ion will be pushed inside the parietal cell with the application of ATPs. So here you can see that's why we need one hydrogen potassium ATPase. So that pump is hydrogen potassium ATPase, where hydrogen and potassium will be pushed out and pushed in with the application of ATP molecule. So these hydrogen ion will be pushed out from the parietal cell inside the stomach lumen, and this potassium ion will be pushed inside the parietal cell. Now once this potassium ion enters inside the cell, then with the application of another unipotar, it will suddenly move out from the parietal cell. So it is a very crucial point where we need to understand what is going on. Basically, after formation of this particular hydrogen ion, these hydrogen ion will go out from the parietal cell into the stomach lumen with the application of ATP molecules. So when this ATP is used, means to push out this particular hydrogen ion, during that same time one potassium ion will be pushed inside the parietal cell. So once potassium come inside the parietal cell, suddenly by using another unipotar, that potassium ion is directly go back again to the stomach lumen. So in next cycle, when another hydrogen ion will push out, then the same potassium ion will again come in. So there will be potassium rotation. When one single hydrogen ion will go outside the parietal cell, simultaneously the same potassium ion will continuously rotating inside our parietal, rotating inside and outside the parietal cell. So first, this number 1 potassium ion will make an entry inside the parietal cell to kick out one hydrogen ion. Once it come inside, then with the help of this kind of unipotar, it directly go outside the parietal cell. So when the second hydrogen ion, when it will go outside, then the same potassium ion again will come inside. Then again using one unipotar, it will again go back to our stomach lumen. When the third hydrogen ion will go outside, then again same potassium ion will come inside. So this kind of rotation will be continuously going on when one hydrogen ion is moving outside. So finally we have our hydrogen ion. So chlorine ion is already present in the stomach lumen. So finally this hydrogen and this chlorine will make our hydrochloric acid. So this is the overview of the formation of HCl inside the stomach lumen. So I hope you like this content. Thank you.